So thanks for coming to the forestry session. And as Anita mentioned, I'll be talking about, it should be how the 2016 drought impacted New Hampshire's forests. Rich talked about climate variability, so this is a nice segue to what I'll be talking about, except I'll be focusing on trees. Okay, so I'd like to start by providing kind of a broad context for what we might expect the climate to look like in the future based on model predictions. So these two figures here show the expected future climate for spring and summer throughout the United States. And the general pattern that we see is that dry areas are expected to become drier and wet areas are expected to become wetter. If we look specifically at the, there we go. If we look at the Northeast, in the spring we expect to see more rainfall and more extreme heavy rainfall events. But in the summer we expect to see longer, drier periods, more prolonged droughts. So greater climate variability, more total rainfall, but also more extreme precipitation events as well. So this trend was highlighted by the extreme drought that we experienced throughout much of New England last summer. These graphs are from the U.S. Drought Monitor, showing the patterns of drought severity at the height of the drought in mid-September. And we see here the red indicates the more severe drought. So the epicenter was located in southeastern New Hampshire, Massachusetts, and western New York. If we take a closer look at New Hampshire, we can see that the central and northern parts of New Hampshire were not as severely affected by this drought as the southeastern part of New Hampshire. Okay, so the main question that we've been asking in my lab is to try to understand how different northern forest types and species respond to extreme drought, as well as what are some of the mechanisms, the physiological mechanisms that might help us explain why different species respond differently to drought. And we hypothesize that many of these northern hardwood forest species may be especially vulnerable to drought for several reasons. But most importantly, if we look at the kind of the longer term history of this region, drought was relatively rare. If we think about what is limiting growth of plants in this region, it's mostly nutrients and sunlight. Water is generally plentiful during the year. And so as a result, plants probably haven't evolved strategies to survive during longer periods of moisture stress. In general, many of these northern hardwood species, like the sugar maples, the birches, the beeches, they're considered mesophytic, which essentially means moisture loving, and they are classified very broadly as being drought intolerant. But I also want to emphasize that this classification is very general. There hasn't been, precisely because there hasn't been a lot of drought in this region previously, it also hasn't been a common focus of research. So this general classification of drought tolerance doesn't really give us a good idea about how the ability of these species to survive and adapt to a future drier world. So the best way to answer this question is to conduct experiments that push ecosystems beyond their ecological thresholds and beyond the conditions that they've experienced in the past, especially considering that in the future we're likely to see much more severe droughts. So that's really the motivation behind this work. We currently have 2 experiments that are simulating a drought, and they are located in one of them is at the UNH Thompson Farm in Durham, and that's in the region that experienced the most severe drought last year. And then the second experiment is in the Hubbard Brook experimental forest, more in the central part of New Hampshire that experienced a modest drought last year. So to tell you a little bit more about these experiments, so both of these studies removed 50% of the throughfall from the plot. So The through-fall is basically the water that falls below the canopy, and we did this by constructing these gutters that capture the water and divert the water off the plots. We chose 50% because this, based on the historical record of rainfall, that this would simulate a 1 in 100 year drought. And we are continuing the experiment for 4 years. So the intent is to really push the ecosystem to a threshold where we can start to understand how these species can respond and adapt to drought. At Thompson Farm, the dominant species are white pine and red oak, and we have 2 replicate plots that are 30 by 30 meters each. The pre-treatment data were collected, it should be 2015, and then our first treatment year was actually last year, 2016. At Hubbard Brook, the dominant species is red maple, and we have, the plots are slightly smaller, 15 by 15 meters, and this experiment was started a year earlier, so we have 2 years of post-treatment data at Hubbard Brook. These are some of the measurements that we've been collecting. Tree sap flow or transpiration, water use efficiency, which I'll talk about in a moment, soil moisture, and soil respiration. So I'll be focusing on these 4 measurements, but we also collected data on tree diameter growth, litter fall, fine root biomass, and decomposition. Okay, so to determine how much water the trees were using, we measured sap flow, which is the water that moves up the trunk of the tree. And we did this using the heat ratio method, which essentially consists of these probes that we install into the tree. And this schematic diagram shows these probes consist of 3 smaller probes that are inserted into the sapwood, which is the conducting tissue that where the water flows in the tree. And then these probes have, the middle probe emits a pulse of heat every 10 minutes. And the probe above, the upper and lower probe then measure the temperature. So with by looking at the difference in the temperature, we can then calculate the rate that the sap is moving. And then if we know the area of the sapwood, then we can calculate how much total water the trees are using, and then scale up to eventually to the whole stand. So once we've installed these sensors, we then cover them with this aluminum foil to protect them from moisture damage and temperature fluctuations, and every couple weeks we go and collect the data from the data loggers. So here we see the data for white pine sap flow. And this is for the 2016 growing season, so starting in June and going to the early October. The top panel shows the soil moisture here. The middle panel shows transpiration. The black lines are the control plots, and the red lines are the drought plots. And then on the bottom we have the rainfall events. So basically what we see is that over the course of the growing season, soil moisture is declining. We have this period of about 25 days at the end of July and end of August, early September, when we have very little rainfall and the lowest, at that extreme drought period of very low soil moisture, and significantly lower, of course, in our drought plot. And then if we look at transpiration or water use by the trees, we see that the trees in the control plot have much higher transpiration than the trees in the drought plot. And I forgot to mention, you can think of the drought that we imposed on these plots was essentially doubled because we had this extreme drought in 2016. So we actually were able to capture a kind of a super drought last year. So if we then look at what happens when we get to that 25-day extreme drought period, the trees in the drought plots essentially stopped transpiration, basically shut down. In contrast, the trees in the control plots only experienced that almost zero transpiration for a couple days, but still we're transpiring at quite low levels until we get that first rainfall event in September. I did want to just remind myself to mention to you, depending on your frame of reference, just to give you an idea of how much water these trees are transpiring. So at the early spring, they're transpiring about 300 liters per day per tree, and that's equivalent to about 6 kegs of beer or 2 bathtubs. So that's a lot of water from each tree that's being transpired, and it's basically going from that very high amount to almost 0 for a few days. Okay, so this basically we're looking at the same information here except for red oak. And we see overall very similar trends of declining transpiration during the growing season. These trees are growing in the exact same plots together with the pines. But I did want to point out 2 main differences. So first of all, if you look at the drought trees, or sorry, the control trees in the black and the drought trees in the red, they're actually very similar. And even here during that extreme drought period, very little difference between these 2 trees, which unlike the pines, were quite different. And here we see a close-up of just those 25 days of extreme drought. And you can really see here how the white pines in the red have almost no transpiration during this period, whereas the control pines that were experiencing the natural 2016 drought were still transpiring fairly well for much of that period. And in contrast, the northern red oak, very similar transpiration levels. So what we can interpret this as telling us that the red oaks don't seem to be as sensitive to the drought, to changes in soil moisture variability as the eastern pine. The eastern pine is responding in a much more sensitive way to these changes in soil moisture. So we can get some better understanding of some of the reasons why these 2 species are behaving differently by looking at the water use efficiency, which provides us with an indicator of drought stress. So just to explain a little bit more about this, so on this, trees have, or plants have, these small openings or stomata on their leaf surface that open to allow CO2 into the leaf, where it can, where the plant can then photosynthesize and produce carbohydrates and energy. But at the same time, when these stomata are open, they also lose water. So there's a trade-off between photosynthesis and water loss. So to photosynthesize, trees lose water. When conditions are dry, the trees can close their stomata and conserve water, but that also means that they can't photosynthesize. So that, again, there's this trade-off that we can quantify as the water use efficiency, how much water is lost relative to how much carbon is gained by the leaf. And we measure it with this instrument in the field. So let's look at the data for water use efficiency. This graph is comparing, we have red oak here and white pine. The green bars are the control trees, and the red bars are the drought trees. So we can see that with red oak, there really isn't much difference between the control and the drought trees in their water use efficiency. So that's basically saying that their stomata are staying relatively open, and they're not closing their stomata to try to conserve water during the drought. The white pine, we see that the drought trees actually have much greater water use efficiency than the control trees. So that's telling us that the white pines in the drought plots are closing their stomata, trying to conserve water. Again, they're more sensitive to those drying soils than the red oak, which is able to continue photosynthesizing during the drought. Okay, so I now want to move to our Hubbard Brook experiment, where we saw a very different picture. So here we see sap flux or water use by the trees during the growing season. And essentially we didn't see, oh, I'm sorry, this is for red maple. We really didn't see much difference between the control trees and the drought trees in these plots. Overall, very similar. We're still not sure why we didn't detect a difference, but we have some hypotheses. It's possible that, as I mentioned, in central New Hampshire, the drought wasn't as severe. But still, we droughted these plots for 2 years. So it seems like, and we did detect a change in the soil moisture, so it seems like we should still be able to detect a response. Another possible explanation is that our experiment wasn't really effective at creating a drought. These plots were smaller, and also they were on slopes, so it's possible that there may have been some lateral flow of water getting into the plots, or that trees had access to deeper groundwater. So this coming season we'll be measuring groundwater and trying to get a better idea of where trees are getting their water from. And and the third reason may be that red maple is actually known to be a highly plastic species. So it means that it can tolerate, or it's found growing under very different conditions. So for example, here in New Hampshire, we find red maple on ridgetops where soils are very shallow and tend to be drier. But we also find red maple swamps where soils are saturated. Maple grows across a very broad geographic region from Maine to Florida. So it doesn't seem to be very picky. And so consequently, maybe red maple is more drought tolerant or can better adapt to drought. This has relevance for the sugar maple industry here in New Hampshire and New England, which is very important economically and culturally. Red maple is also used for sugaring, and we know that sugar maple is a very sensitive species, a drought-sensitive species. So this may provide an alternative sugar maple species in the future. Okay, so finally I wanted to just show some results from soil respiration at Hubbard Brook, because this was actually the only variable that did respond significantly to the drought. We saw lower soil respiration in the drought plots compared to the control plots, which tells us that the soil microbial community may be more responsive to the drought than the trees in the overstory. And we have some theories on why that may be that we'll be looking at. So in summary, so far our preliminary results suggest that white pine is more sensitive to drought than red oak. We also see that red maple may have a greater capacity to tolerate or adapt to moisture stress. Because these trees may take a long time to actually respond to drought, it's really important to conduct these long-term studies that can actually push ecosystems beyond thresholds. And these experimental drought manipulations provide a useful mechanism or tool to understanding how ecosystems are going to respond to future climate change. So with that, I'd like to thank the many collaborators on this project, as well as the landowners and managers who we've been working with, and the funding sources. And if I have questions, I'd be happy to answer questions.